Alright guys, so this is our lecture for classification and evolutionary relationships. You might want to have your journal out while you're going through this, just because I will be checking journals on Monday or Wednesday. So make sure that you fill out your chart. So the first thing we'll talk about is what is the definition of taxonomy, and that's just the science of naming and classifying organisms. So when we give something a Latin name that helps us identify the organism, we're taxifying it. Okay. Aristotle was the first scientist to start grouping plants and animals based on similarities. These were actually there's physical similarities because at the time they didn't have the technology to look at DNA similarities or anything that was beyond the naked eye. The Greeks and the Romans actually gave the name genus, which means group. So they were the first ones to kind of give a name to those major groups. And then the most famous scientist, Carl Linnaeus, he was Swedish and gave organisms a two-word name, and this is what we now refer to as binomial nomenclature. So that's just a fancy word for a two-word naming system. So bi means two, nomial means name, and nomenclature is just a system. Okay. So it just means a two-word naming system. The genus is the first word and the species is the second word. It's really, really important for you guys to remember that the genus is the first word and the first letter is always capitalized on that, and then when we write the species, it's all lowercase. And then you're also going to underline each individual word, not the whole thing, but each individual word, or if you type it, it needs to be in italics. So kind of the same idea that you're going to do in English when you use a journal or a book or a magazine or any type of name for a literary source. So this is what we're going to call the scientific name. So if you guys ever hear me refer to a organism by its scientific name, you know that on your ID sheets I always give you the scientific name and you get extra credit for writing them correctly. This is how you do it. The genus is the first word, first letter is capitalized. Second word is the species, it's all lowercase, and they're both underlined individually or written in italics. But don't write them in italics because it's really hard to do that. It's just, you need to, if you type it, anything, it needs to be in italics. So our universal language is Latin, and I think we kind of talked about this a little bit and touched on a little bit that it's a standard for communication because it's what we like to call a dead language because it doesn't change over time. That's really important, but it's also important because there are so many Latin words that don't change over time. We can give every organism a different name, so you won't have any organisms with the same two Latin names. They can share one or part of their name with another organism. So an example of that would be like Quercus alba is the white oak. Quercus falcata is the southern red oak. So they share the same genus name, but they don't share the same species name, which indicates that they're different species in the same genus. So when we look at our hierarchy of classification, most of you probably know how this works, but it starts out with domain, then it goes to kingdom, and then phylum, then class, order, family, genus, and then species. So you guys can kind of come up with a silly, like mnemonic device that helps you remember this. Usually, most people will use Dear King Philip came over from Germany swimming. You can come up with anything you want. Dear King Philip came over for good soda or grape soda or whatever. But remember that the genus and species are going to be your scientific name. Here's one that, this one is not mine, but this is one you can use, or you can come up with your own. A domain is just the highest order that you can have. It's the most broad. And then as you go down the list here, it gets more specific. So let's talk about species. The two most important ones we'll talk about is genus and species just because they are part of the scientific name. But they're generally defined as a group of organisms that can breed and produce fertile offspring. Most dogs can interbreed with one another. Types of dogs can interbreed with one another and produce fertile offspring. It'd probably be pretty hard, but Great Danish Chihuahua can breed and have fertile offspring. because they're both Canis familiaris. They're just different breeds of dogs in the same species. Horses and donkeys are different species, and when they breed, their offspring are sterile. So most of you, we live in a farming community, so most of you probably know that mules can't have babies. So they're the same genus, but different species. So we're going to talk about the 6 kingdoms. This is how you're going to fill out your journal. So make sure that you have that out so that you can start filling in your characteristics. Remember that you need to have a couple of characteristics in the box, and then in the smaller box in the bottom, and you need to have a drawing of what type of organism this is. So Archaeobacteria, Eubacteria, Protists, fungi, plantae, and Animalia. These are all of our kingdoms. But recently, they've been starting to put Archaeobacteria and Eubacteria together and calling them Monera. So some people use a 5-kingdom system, some people use a 6-kingdom system. Just remember that if we're using a 6-kingdom system, it's just splitting up the bacteria, the Monera, into Archaeobacteria, which is ancient bacteria, and then Eubacteria, which is true bacteria. So I'm going to go through this pretty quickly because most of you know the difference between an autotroph and a heterotroph, but an autotroph makes its own food. Auto means self, so if you think about an automobile, it powers itself with its motor. The autonomous nervous system is the one that does all of your involuntary responses like blinking, breathing, digesting, all of those things. So auto means self, so this is a self, it makes food for itself. They're usually photosynthetic. We all know that photosynthetic organisms use sunlight to make glucose, just so they can get food. And then we also have chemosynthetic, which means that they can take nutrients from chemicals, or actually just elements like sulfur, iron, to make food from that. But all autotrophs are going to make their own food. So plants, and then chemosynthetic bacteria. Okay, and then heterotrophs are just the organisms that can't make their own food, and they have to eat other things to get food. So they either have to eat other organisms or the waste from those organisms, like decomposers. And then you also have absorbers. An example of absorbers would be like a mushroom. They absorb nutrients from dead and decaying wood or leaf litter to make into their system and digest those molecules. So we'll start out with Archaeobacteria. So Archaeobacteria is going to be things like halophiles, which are salt-loving bacteria. You've got methanogenic or methanic, which usually are hanging out in cows' digestive systems, because that's where you get a lot of methane. And then you've got sulfurous, which that's where you're going to find these types of bacteria in like hot springs and things like that. So you can see that we don't have a whole lot of them. There are only a couple of branches, so they really haven't evolved much from the beginning of time, pretty much, on Earth. So there's not very many of them, but they do thrive in extreme environments. Okay, they're prokaryotic, and if you remember from biology, prokaryotes are organisms that don't have a nucleus, and they don't have usually don't have many organelles, save maybe some ribosomes and some genetic material. So just remember that these are your bacteria, which are usually going to be prokaryotic organisms. Archaea just means ancient, so these are these ancient bacteria that have been around for a really long time. It's sort of thought that a lot of our living organisms that we have today have evolved from Archaeobacteria originally. They don't have a nucleus, no organelles, but they do have a cell wall, they have a plasmid, they have lipids, so they do have some things that normal cells do have, they're just not as complete. They're unicellular, they can be autotrophs, heterotrophs, but they really like those extreme environments like volcanic vents, hot springs, salt flats. So we actually have lots of those halophiles, which are salt-loving bacteria, Archaeobacteria in the Great Salt Lake, which makes it pink, sort of, if you see it from like a plane picture or something like that. They usually like areas without oxygen, which are anaerobic or anoxic, and they reproduce asexually, which is usually how most bacteria are going to reproduce through binary fission. And there's actually less than 100 species, so if we look back on the last slide, we notice that there weren't as many branches. So here are some examples. We've kind of already gone through these: halophiles, acidophiles, sulfurous bacteria, methanogenic, anaerobic bacteria. So anything that kind of lives in an extreme environment would be an example of archaebacteria. So eubacteria, this is just everything else that's in that bacteria. They're prokaryotes, which just means that they still don't have a nucleus, but U means true, so these are your true bacteria. These are going to be things like E. coli, Staphylococcus. There's also good bacteria like, it'll come to me in just a minute, but the ones that are in yogurt that help your digestive system. And we do have to have bacteria in our digestive system to create vitamin K, which is an essential vitamin that we don't get any other way. They're also, since they're prokaryotic, they don't have a nucleus or organelles. So kind of the same as the archaebacteria, they're just, there's just more of them. They have the same type of feeding strategies, autotrophs. Some of them can be heterotrophic, but they live everywhere. I mean, right now, you've probably got tens of thousands of bacteria on your hands, even if you just wash them. So bacteria pretty much live in every environment on our planet. And they're very diverse, so over 5,000 species. Some of them cause disease, some of them make vitamins like vitamin K, what I was talking about. Some are decomposers, some fix nitrogen, so they have a lot of different niches that they kind of have evolved for. Some are good, some are bad, but bacteria really are essential in our lives to drive evolution, but also to help us create nutrients that we need and help our immune system. So strep, cyanobacteria, salmonella, which you get when you get food poisoning, botulinum, which is botulism, and then E. coli, which we need some of in our system, but if we get an overload of E. coli, it can make us sick. All right, so here are eukaryotic. Here are eukaryotic kingdoms. So Protista, fungi, plantae, and animalia. You can see that there are a lot of them. We're going to go over them pretty quickly. Pause if you need to, write some facts down. But for the most part, you don't have to write down a whole bunch, just, some main points that are going to help you a little bit. So we'll talk about Protista. This is kind of the catch-all kingdom. This is where we put things that are photosynthetic and usually unicellular that we really don't know where to put anywhere else. So they're eukaryotic, which means they have a nucleus and they have, organelles, and they function just like a human cell would. They're usually going to be photosynthetic, but they're mostly unicellular. So things like algae, amoebas, dinoflagellates, things like this, a lot of them are going to be hanging out in the ocean. They produce about 1/3 of the world's oxygen in our atmosphere. They can be from really big to really, really tiny. They have many modes of reproduction. The main thing that is really important about this is that they're normally going to be photosynthetic. They're going to be unicellular, and they need water in their environment. So those are the 3 main things that I want you to remember about this category. And when they're photosynthetic and there's lots of them, then we can kind of logically assume that a lot of our oxygen comes from this category. So that's probably another thing you might want to write down, is about 1/3 of the oxygen in our atmosphere comes from these guys. All right, so fungi, most of you probably know mushrooms, but there are also a lot of other things that go into this this kingdom. So they're eukaryotes. So again, they have a nucleus and all of these organelles that help the cell function. They don't have chloroplasts because they're not photosynthetic. They don't make their own food. They're absorbers. They actually get their nutrients from dead and decaying material like wood or leaf litter or even manure, but even dead and decaying organisms. They have the cell wall that's made up of, that's kind of what gives them that milky white kind of look to them. Some are unicellular, but most of them are going to be multicellular. Usually the multicellular ones are going to be like your mushrooms, your sporolites, your mycorrhizae, but your unicellular ones can be those molds. Okay, so they're heterotrophic because they use absorption, but they're mostly decomposers. That's why you're going to see them in like the leaf litter layer of a forest, because they're taking that decaying leaf litter and recycling it back into the nutrient system. They're terrestrial, which means land, and some of them are actually going to live on other organisms. They reproduce asexually through spores, usually. Some can cause disease. Some fungi can cause disease. I mean, y'all know about foot fungus, okay? So some of them can cause disease, but a lot of them do a lot of good for us. We eat them. They fix nitrogen for plants. We can use them to make medicine, but they can also be poisonous. So don't eat them unless you know what it is. And even if you do know what it is, maybe you don't know what it is. So I strongly suggest that you don't eat mushrooms unless you buy them from the store. Bread molds are an example of this. Mushrooms, yeast, mildews, truffles are a type of mushroom, really, really expensive one that you probably will never get a chance to eat unless you have like thousands of bucks. Penicillin is also a fungi. It's what we use to help get rid of infections. Some people are really allergic to it, some people aren't, but it was, when penicillin was found, it was a really great advance in our medicine. So plants, or Kingdom Plantae, you can see that we've got about 4 different kinds of plants. You know, you got your bryophytes, which are your mosses. You've got your vascular seedless plants. Those are things like ferns, because they have those spores underneath their leaves. Gymnosperms are going to be your conifers, like your pines. And then your angiosperms are any type of flowering plant. So they're eukaryotes. We all should know that. We know that they have a cell wall, they have chloroplast, they're autotrophic, they make food for themselves, but they have cell differentiation, which gives them roots, leaves, reproductive structures that are different than humans. Okay, they're multicellular. We don't have anything in the plantae that's going to be multicellular or unicellular. Excuse me. They're terrestrial usually, but we do have some in the water, like sea kelp, and we have mangroves that kind of live in salt marshes, so we do have some that can be water-based. They have sexual reproduction through sporulite, so they can send out their pollen or their pollen, and we need pollinators for that. They provide oxygen, they convert energy, and we use them for a lot of things in our textiles, medicine, so plants are really essential for our growth as humans. And you guys can probably give yourselves some examples, but trees, flowers, ferns, mosses, grasses, so anything that's autotrophic, that's multicellular, is usually going to be in this plantae family. Okay, so Animalia. Animalia is us. You know, we're eukaryotes. We don't have chloroplast or cell walls, but we do have cell membrane, and we have vacuoles and mitochondria and all sorts of things that help us differentiate our cells. We have complex organ systems, multicellular. We have to eat things to get our energy. But Kingdom Animalia lives just about anywhere. We can reproduce, well, we can't, but Animalia itself can reproduce sexually and asexually. There's a couple of different ways. But, you know, we use a lot Animalias for food, clothing, all sorts of things. So this kingdom is really important, not only because we're in it, but also because we use other animals that are in it. So here are some examples: snails, sponges, fish, turtles, snakes, mammals. So pretty much anything that is multicellular, heterotrophic, and is usually going to be a chordate, but sometimes you have these, you're going to have invertebrates, and also some annelids. All right, so let's talk about, now that we've kind of gotten out of the characteristics of our different kingdoms, let's talk about how do we put things into those kingdoms. So a phylogenetic tree kind of gives us a diagram that represents relationships between organisms, and we can use this to find clues to relationships to one another. A really good way to kind of look at this is fossil records. So we're looking at, do we have things that are similar in our fossil record? Are they in the same kind of geological strata? What about the correlation of their DNA? Can we get DNA from the bones? Can we radioactively date them? So we can kind of get an idea of like a geological time structure of where these organisms were found and if they're related to one another. Now, this is just one of a few ways that we can kind of figure out if organisms are related to one another. This is a cladogram, and we're going to talk about cladograms in just a minute, but you can see how closely related things are to one another. So you can see that crocodiles and birds are actually more closely related to each other than birds are to mammals, or crocodiles are to snakes. So this kind of gives you an idea of how things, when things are classified, we're looking at that phylogenetic tree, we're looking at the DNA correlation, we're looking at that geological time scale to kind of see what's related to what, because sometimes just looking at it physically, it may look like something else, but they may actually not even be related to each other, except for maybe a common ancestor thousands and thousands, millions of years ago. So it's really important to look at all of these things and factor everything in to kind of get an idea of whether they're related to one another because they look similar or because maybe their DNA structure is similar. So a dichotomous key is something that we use in science. If we're looking at a plant or an animal or something and we don't know what it is, then we can look at this key to kind of help us figure out what it is. It's kind of like one of those books that you read as a kid where you got to choose the ending. This is kind of the same idea, except for you're going to be following the instructions at the beginning. So it's just a key used to identify something, and you're just going to use pairs of contrasting descriptions. It's going to lead to another pair of descriptions or identify the object. You're going to start at one every single time. So if you have a tree in front of you and it has needles, the first one may say, does your tree have leaves or needles? And you're going to say, okay, needles. And it's going to say, okay, go to page 120. You're going to go to page 120, and that's going to ask you another question. It's going to direct you somewhere else until you get to that identification of that organism. So here's another cladogram. They're just diagrams that show evolutionary relationships between a group of organisms. The 4 traits that are listed on this diagram are jaws, lungs, claws or nails, and fur and mammary glands. Okay. So organisms that are below that derived trait. So if we look at jaws on this, that means that anything that's below that doesn't have that. So hagfish don't have jaws. Perch don't have lungs. So if it's below that, it means that they don't have that trait. If it's above it, it means it does have that trait. So we can look at this cladogram and say, okay, what traits do pigeons have based on this cladogram? So you know that they have claws or nails, lungs, jaws, and then they also have feathers because that's just on their line. But they don't have fur or mammary glands because, first of all, they're not mammals. Okay, but it's also because pigeons are below the fur and mammary glands trait line. Okay, so the last couple things I want to talk about is kind of goes along a little bit with technology and classification based on physiological traits. So organisms can be grouped into categories based on physiological traits. Okay, and that physiological traits are going to be things that you can actually see with the naked eye. So an example of that is going to be mammals that have mammary glands. And so all of those organisms that are going to have that physical trait are going to all be classified as mammals. Now, if I go out and I find a brand new organism and it has a mammary gland, that doesn't necessarily mean that I'm going to put it in mammals because I'm going to have to look at the other traits, but that can at least give me an idea of that's probably where it's going to go. So I'm going to use this in addition to other things. So those addition to those other things that we're kind of talking about is the technology involved. So genetics are really the best way to group and classify organisms because we can look at the genetic code of an organism and classify it as closely as we can with another organism that may have the same type of genetic code as this one. So technology advances all the time in the genetic field. So as the technology gets better, classification can get more accurate. And you'll see sometimes where organisms that were classified in one group before may be reclassified in a different group based on the advances in technology. So we have to kind of keep that in mind that sometimes organisms can change classifications because of the advances in technology. You know, the genome projects of organisms reveal new traits. We are sequencing more and more animals and we're knowing more and more about them and their traits, and so that helps us classify organisms as we discover them or as we regroup them. You know, a classification of organism, a family, a genera, even a kingdom can change, which seems kind of crazy, but, you know, it happens. But as we advance in technology and then also this carbon dating, we can accurately discover and classify fossils, which can help us regroup things based on what we find in the fossil record. Okay, so as technology gets more accurate, the classification of organisms can change. But classification has changed even from the time of Linnaeus. So as we find more accurate classification methods, those are going to change over time. They're not going to stay the same. They're going to change and be dynamic over time. Okay, and we have to think about the invention of the microscope. Once we were able to see things beyond the naked eye, we were able to reclassify organisms. We were able to even add 2 more kingdoms, Archaeobacteria and Eubacteria, because we could see them. So it's just really important to remember that technology advances, and these advances in technology really give us an idea of how and why we're classifying things. Okay, so new extinct species can be recovered using carbon and argon dating because we find fossils, fossils we've never seen before, and so those certain species can help us in relation to newly discovered organisms. So maybe something will change based on a newly discovered organism that we find. You just never know. So it's really important to kind of keep up on that kind of stuff because things can change, and it's just a really good idea to kind of keep up with that. Okay, so remember to fill out your journals, and I will check your journals either Monday or Wednesday when I see you. So please get this done. See you guys later.